Hey, hi, good morning, everyone. Um, so this is our second to last lecture for the semester. So this week, you still have your last recitation meeting with your TIs. I hope it's a good one, right? So you have your last homework assignment due this week in recitation. I've actually decided, previously I had mentioned I thought I was going to give you one more homework assignment that would give you a way to kind of work on some of this week's material, like the cell cycle stuff, cytoskeleton, meiosis. But then I realized, I know everybody has a lot going on at the end of the semester. So I've decided not to give another homework assignment, okay? So the homework assignment that's due this week in recitation is your last graded homework assignment. What I will do is I'll give you a worksheet that has like homework-like questions on it that is entirely optional for you to use as you study for the exam. All right, and that optional homework assignment sheet will have stuff on the cell cycle and the stuff that we're talking about this week. Okay, so make sure as you finish this week's homework assignment, do a really good job because it's your last one, right? So you want to end really well. And also this week, of course, is your last quiz. So also study really hard and do a really good job on your last quiz as well. And of course, try to enjoy your last proper recitation section with your TI. I was talking with the TIs in our TI meeting this week, and I told them, yeah, this is your last meeting with your students, even though next week you'll still be meeting with them, but it's not going to be very happy circumstances because that'll be when you're taking the exam, right? So anyhow, make this week's recitation a good one. All right, let's talk about meiosis. So when we were discussing the cell cycle and when we were talking about mitosis, right, mitosis is the process in which a cell in your body, a typical somatic cell, meaning any of the cells in your body that are nucleated, that are not the cells that are involved in reproduction, right? So the non-gamete cells, any of the other cells in your body are often referred to as somatic cells, right? And these are the cells that are carrying out their different jobs of making all the complex different types of tissues, organs, systems within your body, right? And so those fully differentiated cells, as they divide, right, they want to make a perfect copy of themselves. Because as you have, for example, a skin cell that's trying to divide so that you have enough skin cells, you don't want to mess with the actual genome, right? The set of genes that they have. I mean, that set of genes in that sort of parental cell represent all the genes that you were born with, right? At the moment of conception. This was the set of genes that you got from mom, the set of genes you got from dad, and that has been your master blueprint your entire life, right? So all the differentiated cells in your body, when they divide, they make sure that the two daughter cells that come from a starting cell have the exact same version of that genome, right? Because you don't want to mess with it, right? However, the gamete cells in our body, right, the sperm and the egg, the oocytes, right, those are different. Those are the process of not just mitosis, but an additional process known as meiosis. Okay, why? It has to do with the fact that in that case, right, when those cells divide and you're producing gametes, egg and sperm, right, the genomes contained in those are no longer the full complement or the full set of a diploid genome that represents what came from mom and dad, right? These are now divided in half so that when they recombine, right, the gamete that you make in your body and the gamete that your mate makes in their body, when those cells combine, you reconstitute a diploid genome. Right? And consider this special opportunity of cell division that happens when making gametes is also the opportunity to kind of mix and match different genes, right, that came from your parents and have been part of your blueprint your entire life, right? What you pass on is sort of a mixing a different recombination of those genes that you got from your parents. And that's what you pass on and contribute in your offspring. So that's a pretty important process when you're talking about organisms like us that reproduce sexually, right? Male and female. And that's why meiosis is really worth understanding and appreciating. Let's start off by thinking about the fact that we have a diploid genome. Remember, ploidy, funny word, right? Ploidy means how many copies of an organism's genome are actually present in a typical cell from that organism, right? So a typical differentiated cell in our body has 2 copies of the human genome, right? One from mom and one from dad, right? So therefore we have 23 pairs of chromosomes or 46 individual chromosomes, right? Representing 2 copies of the human genome. And that's why we are diploid. So what are some of the benefits of being diploid? Well, if you have 2 copies, right, of each chromosome, then you have more flexibility in how to express the products of certain genes, right? So if you have, let's say, a gene that you need to produce a lot of, right, then maybe you can utilize both copies of that gene, right? Both the chromosome you got from mom and dad that have that gene on it, right? Maybe in that case, both copies of the gene can be expressed to make high levels of protein product. Another important thing to consider is every day we are subject to random mutations, mistakes that happen in our DNA sequence. Right? If you get a mistake that kind of messes up one copy of your gene, let's say the one that you got from dad, right? The fact that you have another good copy from mom might be good enough, right? That your cell can still make the proper protein product or whatever it is that's being made from this gene, and you're okay, right? So having a good backup copy is another benefit of having a diploid genome. And the fact that the copies that you get from mom and dad, even though they are the same chromosomes containing the same genes in the same arrangement on those chromosomes, right, the actual nucleotide sequence of the different versions of the gene that you got from mom and dad might be slightly different based upon ancestry. Right? And therefore having not just 2 copies of this gene, but one that's ever so slightly different than the other, right, might mean that the protein products from those genes are a tiny bit different from each other. And you never know, depending on natural environment, selective pressures, if one version of the protein actually is more beneficial or useful than the other version, right? As things in the environment change and put pressure on our species, right, to select for certain changes, mutations that actually are beneficial, right? But with these benefits come challenges, right? Because, hey, if we have a diploid genome, that means we have twice as much DNA in each of our cells, right? 2 copies of the entire genome. That means there's way more chance of random mutations happening if you have double the amount of DNA. Right? Some of these random mutations might be really bad and mess up a gene in a way that the expression of that gene or the activity of the protein that comes from that gene, right, is no longer controllable by the cell and causes all kinds of problems like diseases. It may also be that if you have detrimental mutations in one copy of a gene, but not the other, and the detrimental mutation isn't that bad because you still have the other copy, then as an organism, you might still survive and have offspring, right? And therefore, you'd actually be passing on that bad version of the gene onto offspring, right? So that's detrimental. And as we already mentioned, the fact that you're diploid, right, means you have to undergo special processes to make reproductive cells that are haploid, right? That represent not 2 copies of the genome, but just 1 copy of the genome that can then join up with the gametes of your partner, your mate, who also provides the other copy of the genome to make a new diploid fertilized egg, all right, the start of a new human being. So that means because we reproduce sexually, there is this extra challenge, right? That the special cells in our body that handle reproduction have to do this meiosis. So, but the bottom line is this, right? Regardless of our sort of human-centric view of a diploid genome being normal, right? We have to kind of stop being so sort of conceited, right? And actually realize that there's no hard and fast rule in Mother Nature that says organisms have to have a diploid genome. No. In fact, all throughout nature, all through the diversity of life on this planet, we see a wide range of ploidy. Right? Everything from simple organisms that are haploid, that just have one copy of the chromosome. And in many ways, if you think about it, you would imagine life probably started off that way, right? Just putting together a usable genome is challenge enough. Chances are life started off with just one copy of a genome in one cell, right? Some organisms are multiploid or polyploid, right? Meaning they have multiple copies beyond just two. Some organisms, for example, like plants, can from generation to generation vary their ploidy. So our friend the carrot, which we kind of used as a couple of examples during the semester, right? Carrots can vary anywhere from having a generation of carrots that are diploid, right, to generations that are haploid, to generations that are polyploid, up to like 7 copies of the genome in a particular carrot cell, carrot individual, right? And it's okay because they have other mechanisms, right, for making that work. So you might say, well, okay, but for mammals like us, diploid seems to be the norm, right? A copy from mom, a copy from dad, and we're all good, right? We stay being diploid or 2N, right? But there is an example of a mammal that is actually not diploid. I don't know if any of you know this, but there's this little rat that lives in South America, in Argentina. It's the Vizcacha rat. And it is the first example, as far as I know, of a mammal that is actually tetraploid, right? Meaning it has 4 copies of its genome in each of its cells, right? Very interesting, right? Because we're not quite sure what to make of it yet. It might actually represent, right, the further evolution of the mammalian lineage of organisms on our planet. All right. So meiosis is not only important for being able to split the diploid genome in half so that you only contribute 1 copy of the genome as part of your sperm or your egg, right, to join up with the other copy coming from your mate, but meiosis is also the basis of genetic variety. Right? Sexual reproduction allows a mixing of parental genes. It's Mother Nature's way of experimenting, right, and trying new combinations of genes against the pressures of natural selection. Right? This is the basis of evolution. Right? Mutations that happen that can be passed on if they are beneficial, right, or eliminated if they are detrimental. And the ability to mix different combinations of genes from parent to offspring is why, right, we have genetic diversity in our species. So not only must the egg and sperm cells carry a haploid or half version of the genome, but that half can't just simply be, right, in some of the gametes that I make, I'm just going to give my kids the version of the genome that I got from my dad, right? Or I'll give my kids the version of the genome that I got from my mom, right? And the same thing with my wife, right? Because that won't really work. Because if that were the case, if you think about this, right? If you have a diploid genome, one version that you got from mom, one that you got from dad, and all you did was simply split that in half, right? So some of your gametes have moms, some of the gametes have dads, and then you pass that on. That's not much variation. If that were the case, this is kind of wacky if you think about it. A couple could only have sort of 4 versions of children, right? the fifth one would be like an identical copy to one of the older siblings. And clearly that's not true, right? The exception is, of course, when you're talking about identical twins, right? Identical twins, but that's because of just the fertilized egg just undergoing division early on and having identical genomes in it. All right. Therefore, meiosis must be really important then, and importantly, it's critically different than mitosis. And I kind of joke here. I say that meiosis is not just some weird version of mitosis that you have to learn in biology class, right? It actually is really important that you understand why we do meiosis, or why the reproductive cells in our body do meiosis. Okay. Let us think about the appearance of chromosomes at different parts of the cell cycle. Remember, interphase refers to all parts of the cell cycle other than mitosis. So when the cell is in G1 doing its normal differentiated cell functions that it's supposed to do, right? It's expressing those particular sets of genes from the master blueprint that are useful for being a nerve cell or a muscle cell or a hair follicle cell, etc. Right? Remember, if you were to look at its nucleus, which is what we see here, you would see that all the chromosomes are more or less unraveled, right? We said this is like a big bowl of saimin, right? Big bowl of noodles. And if you could actually figure out where different chromosomes are bundled up, you would sort of represent it like this, right? The little dotted lines are the boundaries between different chromosome territories. And within each of these, the gray area is just the long chromosome itself, all unraveled like a noodle. Parts of it might be more euchromatic. So the light gray regions represent parts of the chromatin that are really unraveled. So this is like the noodle is really unraveled. And you might imagine that along that stretch of the chromosome are genes that the cell needs to have access to at this time, right? Maybe those are genes that are useful for things like metabolism, or for certain types of making certain types of proteins that this particular type of differentiated cell needs to worry about, that maybe other types of differentiated cells couldn't care less about, right? And you have regions of the chromatin that are highly condensed, heterochromatic. All right, so these would be these darker regions, where in this particular cell, the genes that are contained in those lengths of DNA maybe are just simply not relevant, right, or not needed right now. Right, so as a result, the chromatin is much more compact, right? It has an important way, remember, of controlling that those genes don't need to be expressed. So we can think about an interphase chromosome as being like this jump rope here, right? So if this is one chromosome, you would imagine that along the length of this jump rope, right, you'd have different genes, right? Now keep in mind, the jump rope itself, as you look along it, doesn't look any different from one part to the other part. Just like our chromosome itself, all it is a big string of DNA, right? A's, T's, C's, and G's. It's not like somehow magically one part is highlighted, like this is a gene or something like that, right? What we have learned in this class is the definition of what a gene is a length of that DNA that actually goes ahead and codes for something. For example, codes for an RNA transcript that then can get processed into an mRNA that can then be in the cytoplasm translated into a finished protein. And remember that different sequences surrounding genes are the regulatory regions like promoters, or distal regulatory regions like enhancers or insulators, silencers, right, that bind to different transcription factor proteins that ultimately communicate with the promoter region of a gene to assemble things like RNA polymerase II and get it to transcribe that particular region of the chromosome, right, to make an RNA transcript. So let's pretend then, along this jump rope, we have, let's say, 3 genes that we care about here. Gene 1 is located over here, gene 2 is somewhere in here, and gene 3 is somewhere over here, right? And when they get expressed, They get made into a primary RNA transcript. That transcript gets edited into an mRNA. The mRNA is then translated by a ribosome to make protein 1, protein 2, and protein 3, right? So these are important proteins that are part of the human, our human cell physiology. Okay, so here we have the jump rope, right? So I think it's important to remember that depending upon where in the cell cycle you are viewing the appearance of the chromosomes, they're going to look very different, right? So at this point during interphase, right, the jump rope is just kind of loosely kind of bundled up, right? It represents one of these sort of gray chromosome territories in the upper part of the slide there, right? And along it, you have those 3 genes somewhere in here that are being transcribed. Right? So that would be, for example, if this were a fully differentiated cell, just happily minding its own business in G1, right? Doing its job, making these proteins so it can be the best skin cell, nerve cell, hair follicle cell, or muscle cell that it can be, right? But if the cell receives the message that it should, not do this right now, and instead think about dividing, right, for mitosis, then it's going to stop doing all the normal gene expression it normally does, right? And it's going to duplicate itself. It's going to replicate all the chromosomes, including this blue jump rope, right? So that would happen during S phase. So during S phase, the jump rope is still pretty loosely organized in there, right? But what would start happening is you'd start seeing all those little origins of replication as the double-stranded DNAs begin to separate, right? And you have things like DNA polymerases making another copy of this particular chromosome. So at the end of S phase, you would not have one blue jump rope, you would have 2 blue jump ropes, right? And that's the importance of S phase. And that's why once the cell starts doing that, it cannot just decide, you know what, I don't want to do this anymore. I want to just go back to S phase because I miss being just like a normal nerve cell or gland cell, whatever. Because what? You're going to have the wrong number of chromosomes now, because some of them are going to be, some of them, if not all of them, right, will be duplicated. So at the end of S phase, you have 2 jump ropes, 2 copies of each chromosome that you have. Right? So as you enter mitosis, that's when you start seeing these chromosomes really condensing now. Right? And so they stop being just like the little jump rope here. They start being like these pool noodles. Right? So each one of these pool noodles, let's say this was the original jump rope, it is highly condensed now. Right? But importantly, what? In here is still gene 1, gene 2, and gene 3, right? But now they're just no longer accessible because they're all bound up in this tight sort of rod-like structure, right? And importantly, remember, you have not just one blue jump rope because you replicated your DNA. When all the chromosomes condense, you find out, oh, you actually have 2 of these blue pool noodles, right? Because one was the original one was the duplicate, right? We call each of these chromatids. So you have a pair of sister chromatids. And that's why, right, you'll see them usually represented during mitosis as this kind of like X-like structure, right? Because they're still attached to each other to make it easier for the cell to kind of organize and keep things straight. So when you look at a karyotype image like this, then that's what you're seeing. Right? So this is taken during mitosis. This is probably late prophase, probably in prometaphase, heading towards metaphase. Right? So remember, our human genome, we have 23 pairs of chromosomes. Right? And each one of these pairs, one comes from mom, one comes from dad. Right? In this particular image, we've already gone through S phase, and so mom's chromosome 1 has duplicated, right? And so these 2 sister chromatids are still attached to each other. Dad's chromosome 1 has also duplicated, so these pair of sister chromatids are together, right? That's why, right, it now looks like this. So not only do you have 23 pairs of chromosomes, right? But the 22 autosomes, each one in this particular image, right, is a pair of sister chromatids. So hopefully you can see that this heads us towards the process of now trying to divide these up. I wanted to point out something that's oftentimes semantically kind of confusing, right? Semantics is, when you talk about different ways of assigning meanings to words and things like that. One thing that I think students get confused a lot about, and oftentimes I still get confused about also, is when we talk about things like our diploid genome as having, 2 copies of the human genome. Oftentimes it's confusing because if you say, well, how many genes are in, well, no, how many chromosomes are in a normal cell in your body, right? You could say, well, I have 23 pairs of chromosomes, right? But you might say, that means I have 46 individual chromosomes normally in a cell that's in G1, just normally minding its own business, right? But another way of saying it is you could also accurately just say, right, the human genome is 22 chromosomes, right, because there are 22 different autosomal chromosome types, right, and a pair of sex chromosomes. Right? So depending on how you word it, all those ways of saying it might be true. It gets even more confusing because you might say, well, how many genes does the human genome have? So our current estimates, so the number of genes fluctuates a lot in terms of what scientists can agree on. Remember, each chromosome is just a long length of DNA, right? It's not like the actual genes are somehow highlighted on them for us to see, right? So you can imagine it's tricky. How do you actually know if a length of DNA is actually used as part of a gene or not? Right? So as a result, current estimates of the numbers of genes in the human genome vary from anywhere from 20,000 genes to 30,000 genes. All right. And also your definition of what a gene is. I've been saying, well, a gene is something, a length of DNA that actually you can detect some sort of RNA product is made or transcribed from that gene. Right? That's one way of thinking about it. Another way of saying it is, well, no, I only want to consider those lengths of DNA that not only make an RNA product, but that RNA product ends up being translated into a protein, right? So you'd then say, well, that is more specific. That's like the number of actual protein coding DNAs. So that still varies, right? Anywhere from 20 to 25,000 protein coding DNAs is what we currently think. So that's why I'm saying semantically it gets confusing, right? Because you could either say there are 25,000 protein coding DNAs in the human genome, right? But that's only talking about one copy of the human genome, right? The one that you got from mom, right? If you were to say, yeah, I have that other set that came from dad, then in reality, I might say, well, we have like 50,000 genes, right? And especially if I just started this lecture saying having a diploid copy is useful because those genes might be slightly different from each other, it's sort of like up for grabs what the most accurate way of referring to the number of genes is. But I think just as long as you can kind of keep it straight as to when you're talking about it in a certain way, what number is appropriate. If you talk about it in another way, what number is appropriate, then you can keep it straight. So think about this. Hey, so I have a little pair of AirPods here, right? And the AirPods are pretty amazing technology. I still think they're amazing. You guys are all probably jaded and they're like, yeah, whatever. That's like a second generation AirPod that Dr. Shen has there. That's old. That's so like 2018, right? But anyhow, I think it's pretty amazing technology. Let's say you were interested in the blueprint or the master blueprint for this technology, right? You could say, for example, wow, there's a lot of advanced technology. I'd love to see the blueprint of what goes into one of these AirPods, right? You could hopefully understand that if I said, well, if I saw the blueprint to make one of these AirPods, this is my left AirPod, then I pretty much know the blueprint to make the right AirPod, right? So I might just say the blueprint to make an AirPod is just this, right? Whatever is needed to make this. However, in a practical, everyday, functional sense, right, these AirPods function as a pair. So you could also say, I need to see the master blueprint for this one as well as this one, even though they're probably almost identical, right? They're only slightly different because of the shape or whatever, right? That's what I'm getting at, right? So if these are the diploid genome in your cells, you could say, right, the human genome, when we talk about it, is just the master blueprint to make one of these, right? Because once you know that, you know the human genome, right? Or you could say, no, I feel like it's more accurate. You need to have both considered at the same time because that's how they work inside of our body, right? The two copies, the one that came from mom or came from dad, right? All right. So let's think about the difference then between mitosis and meiosis. So in this cartoon here, we're going to compare and contrast the two. This comes from your textbook. I'll tell you right off the bat, I don't like the way they draw this in the textbook because this very top one is sort of like we're starting from this parental cell, right? Now, I don't mean parent cell necessarily meaning like this is the cell that came from your parent. I mean, this is the cell in your body that you're starting with in this discussion, right? So before chromosomal duplication takes place, you have a diploid genome. Remember, in these cartoons, to simplify things, we are pretending that the human genome only consists of one long chromosome and one small one, a short one, right? So we'll say chromosome 1 and maybe chromosome, I think chromosome 21 is actually technically the shortest of the human chromosomes. So we'll just say that, all right? And we'll forget about all the other chromosomes in between, right? So you're diploid. So you have one copy of the large chromosome from dad and one copy from mom. You have one copy of the short chromosome from dad and one from mom, right? So inside each of your nucleated cells in your body, this is what you have, right? Now, the reason I don't like the way they drew this is because they say before chromosomal duplication, right? And you can see already the cartoon has drawn these as though they are condensed rods, right? So that's not accurate. Because as we've been talking about, if you're looking at a cell in G1, before any chromosomal duplication has happened in S phase, the chromosomes would actually appear as jump ropes, right? Or as noodles, not as the pool noodles. They would appear as Simon noodles. They would not appear as the big condensed foam pool noodles yet. Right. So during S phase, you'll have chromosomal duplication, right? So you'll have the two copies of the long blue one and the two copies of the long red one, right? They're sort of mingling together. But then during mitosis, they're going to condense into those firm, into those stubby rods, right? So here you see the sister chromatids are still together, right? And we call these sister chromatids, right? Still paired up. During prophase of mitosis is when these chromosomes first become visible as they're condensing like this. Then remember, during metaphase, the importance of metaphase is great. Now you've duplicated all the chromosomes. How are you going to divide them up equally, right, into the two daughter cells? Because if they're all just kind of haphazardly in there in the nucleus, how are you going to somehow cut the nucleus in half or something in a weird sort of windy way in order to make sure that it's evenly distributed? No, it's much easier if you first organize, right, the condensed chromosomes along a central metaphase plate, right? And that's what you see here. So the chromosomes line up and they are organized because you have these microtubules. Remember, we learned about that last lecture. These microtubules made of alpha and beta tubulin that grow from these centrosomes on either side of the cell, right? These tubules grow and they attach to the kinetochore proteins in the centromere of the sister chromatid pairs, and that sort of pushes then all of these pool noodles, right? These thick pool noodles, right? They will get pushed to line up in the middle of the cell along this metaphase plate. Now this makes things easier because once they're lined up, you have one on the left, you have one on the right. Now when the spindle fibers retract and pull the sister chromatids apart, you now have equally distributed them into the daughter cells. Right? And importantly, what? This is mitosis. So a cell that's undergoing mitosis, it wants both daughter cells to be identical in the genome compared to the cell that it started with, right? Compared to the parent cell. And that's what you get. Right? Okay. Meiosis then. Meiosis involves some of these same mechanisms, but importantly, there are two additional considerations you have to think of. Right? One is that you don't want to be left with daughter cells that are like this, because these daughter cells still have a diploid human genome in them. Right? So there must be another division in order to divide these daughter cells further in half, right? So that each resulting gamete cell only has one copy of the human genome in it, right? So that's one difference. But the other important difference, having to do with genetic variation again, is when these chromosomes are lined up, you want to have the opportunity to actually start mixing and matching different copies or different versions of genes from mom and dad, right, together to make sort of like new versions of these chromosomes that can then be passed on. So therefore, meiosis happens in two stages, right? Meiosis 1 and meiosis 2. Meiosis 1, which is pictured over here, is the opportunity for genes to kind of mix. Right? You can see that happens because the way that the chromosomes line up, right, during meiosis 1, during what we call prophase 1, so now each of these will have a Roman numeral after it, prophase, metaphase, anaphase, telophase, right? Will all have a Roman numeral 1 after them. You can see they line up next to each other like this, so that the arms of the chromosomes have the opportunity to kind of mingle and cross over. Right? So we call this formation a tetrad. And during metaphase 1, right, you have these tetrads lined up like this, and you have crossing over taking place. So that now, when these are divided up in anaphase and telophase, right, these chromosomes are a little bit different than what they started as. You can see the pink and blue parts have swapped over. Okay? Let me stop to share for a moment, and I can show you this. All right. So here we have these are, let's say, chromosome 1, right? These are the long chromosomes. And you have one from mom, well, one from mom, one from dad, right? So these have been happily sort of in the cell of your body, differentiated cell in your body. And now it's time, this particular cell, right, is not like the other cells that make up your muscles, skin, nerves, bone, whatever, right? These are cells that are going to become gametes, right? So these are the cells of your reproductive system. So they start off with the genome just like any other cell in your body, right? And what happens is as they condense, right, you would have, this is really hard to do from my desk. This is much easier in person in the auditorium, but whatever, right? So you have your dad's chromosome 1 that made a duplicate, and now they're condensed during mitosis. You have your mom's chromosome 1 that also did the same thing, right? Okay. So if this were mitosis, right, it's almost like you're done, because then these would just line up like this, right? And then the ones on the left would go to the left. That's your right, but whatever, that's my left. And the ones on the right would go off to the right, right? And you end up dividing up the chromosomes evenly. So each daughter cell would again have an identical set of one of mom's and one of dad's, right? So that's mitosis. So meiosis is importantly different because you want to have the opportunity to have genetic material mixed up, right? So during meiosis 1, as these things come together, right, they form a tetrad. So they will actually bunch up like this. Sorry, other way around, like this. Right? And remember, these are all chromosome 1s. So those 3 genes that make proteins that we were talking about on the previous cartoons, gene 1, gene 2, gene 3, I mean, these are the same human genes, whether you got them from dad or you got them from mom, but because of your parents coming from different, families, different genetic backgrounds, the actual specific DNA sequence of each of these genes might be a little different, right? Not necessarily, a million miles different, but different enough, right, that there's some variation. So during meiosis 1, these things come together as a tetrad. And what that enables them to do then is to swap, right? You can swap this part of chromosome 1 from mom with this part of chromosome 1 from dad. Okay? And as a result, what do you get, right? You sort of get new versions of these chromosomes. But importantly, gene 1, gene 2, gene 3, they're still there, right, at the right spot where they're supposed to be, but this is something new, right? It's a new recombined version of these different genes that came from 2 different family lineages. Right? Now, when you divide these up, right, and you make gametes from them, you will be actually making gametes that have different versions of these genomes, right, compared to all the other cells in your body. All right. Yeah, that was way more difficult than it had to be. And now I'm probably going to mess up with how to get back to the screen sharing. So bear with me here. Where did my screen sharing thing go? Where did it go? It like disappeared. Okay. Oh, screen sharing. Here we go. Okay, good. All right, so we're back to this now. All right. I have the chat open just in case I screwed this up. You can let me know if the slides are not working again. All right. Okay. So that's what gets us to this step down here. So at the end of meiosis 1, you've taken advantage of the fact that by forming these tetrads, the DNA molecules can actually mingle. Right? And you think, well, that's kind of weird. How do they do that? Remember, each chromosome is just one chain of DNA, right? So you have 2 chains of DNA kind of like mingling together. It's quite possible, right, that along the way, the chains can break and just reform connected to the other chain. Right? And by doing so, you've now mixed and matched different genes. So from this point on, meiosis 2 is very much just like mitosis. All right. So prophase 2, metaphase 2, anaphase 2, telophase 2, you can see is very similar then to what just happens during mitosis, right? Because now when you divide these up, you end up with 4 daughter cells, each with a haploid complement of the genome, with now these versions of the genes that don't exist in other cells of your body, all right? Because now this really represents the mixing and mingling of the genome that you have, and being able to pass on something unique. That is the source of genetic diversity. Okay. So let's just look at these a little bit more in detail. So again, meiosis, so this is only happening in the cells of your body that make gametes, so only the cells that are part of your reproductive system that make egg or sperm, right? So remember, it's in 2 parts. So in the figure on the left, this first part over here is meiosis 1, right? So meiosis 1 is all about allowing the crossing over of arms of chromosomes to mix different versions of the genes between the 2 genomes that it has in it, right? So early prophase 1, you start seeing the condensation. Mid prophase 1 and late prophase 1, you have them lining up in this tetrad form, right? And you'd have they would form what we call bivalents, right? So that during these bivalents, this is when the arms of the chromosomes can cross over. Right? Then it enters metaphase 1, where it lines up along the middle, anaphase, telophase, and cytokinesis, where they now get pulled apart. Right? So then this is followed by the second meiosis, or meiosis 2, which would be the bottom part of this figure. And as I said before, this is more straightforward, right? So if you've already studied mitosis, you can think of meiosis 2 as just being like, okay, now it's doing mitosis, right? Prophase prophase 2, metaphase 2, where they line up in the middle. Anaphase 2 is now when they're going to be divided apart. And telophase 2 and cytokinesis is going to now result in the 4 gamete cells, right? Each one being haploid, meaning each one having just one copy of the human genome. And importantly, that copy of the human genome that it has is different than the genome that in the other parts of your body, because these now represent each chromosome having an opportunity to swap parts of it with the other paired chromosome, right? So this is a way of putting that together. So during, in males, spermatocytes that are going to form sperm cells, they start off as being diploid, just like every other cell in your body, right? They'll undergo meiosis 1. This is where crossing over is going to take place, right? And you end up having 2 daughter cells here, then that are still diploid. Then during meiosis 2, right, these diploid cells are going to divide to become haploid, right? And so you end up with the sperm cells. Right? And each one of these now carries one copy of the human genome, right? That's different than the genome that you carry, because each of your cells carries a distinct chromosome 1 that came from your dad, a distinct chromosome 1 that came from your mom, right? But the chromosome 1 that's carried in the sperm cell now, that chromosome 1 is made up of some genes from mom and some from dad, right? And that's what you pass on. Likewise, in the female, right, in the women, the developing oocyte in the ovary, right, it also undergoes meiosis 1. In the process of undergoing meiosis 1, as it divides, right, it actually gets rid of the extra set of the genome. You can see here, it becomes just like this little what we call a polar body, all right? And that's going to be discarded. During meiosis 2, right, it's going to then divide again, right, to form now the resulting haploid egg. And importantly, that second division, both sets, both the main cell as well as the polar body divide, right? So you actually end up with 3 polar bodies, 2 that came from the original polar body and the 3rd polar body that came from the second meiosis, right? And what you're left with then is the egg cell, right? 2 things I want to point out to you really fast. One is you might say, well, that's kind of weird, right? Because why don't you just produce 4 egg cells? Like just like in a male, you produce 4 sperm cells from 1 spermatocyte, right? How come in a woman, you don't produce 4 eggs, you produce just 1? This is just evolution. It is because the way we evolved, right, it is more evolutionarily beneficial if we are designed, right, to give birth to 1 child at a time, right? Because, you know, human beings, we're super helpless when we're first born, right? We can't do anything, right? I mean, you can barely even cry until like you're, you know, delivering obstetrician actually like slaps you or something like that, right? Compared to other animals like a giraffe, you know, can get up and start running around in 20 minutes or something like that, right? Or you think about, for example, other animals that are born in litters, right? In those cases, you would have multiple eggs that are fertilized at the same time. So a litter of kittens or a litter of puppies, right? So we evolved, right, to not want to be able to give birth to a litter, because if you did produce 4 egg cells at a time, right, then the default way of reproduction would be, you know, when a woman gets pregnant, she's getting ready to give birth to 4 kids, right? That's not good, right? Because the way we've evolved, right, and the amount of resources and attention that we have to give to a newborn child, right, it works a lot better if you can just focus on having 1 kid at a time to worry about. Okay. The other thing I wanted to point out to you real fast is, you may not know this, right, but the egg, the developing egg, actually pauses during meiosis 2 and does not complete meiosis 2 until fertilization happens, all right? So once sperm fertilize the egg, right, 1 sperm cell manages to get into the egg first, right, and that's what then prevents the other sperm from fertilizing it. Then the developing oocyte will finish meiosis II, jettison the second polar body, and then become an egg cell that's ready now to fuse. The ovum is then ready to fuse its haploid genome with that of the genome being carried by the sperm. Right? And again, the version of the genome that's in this egg, right, is different than the version of the genome carried everywhere else in your body, right, because this represents chromosomes that are the recombined versions of genes from your mom and your dad, right, that you are passing on. Right. Okay, so this is just a neat photo at the end that just shows you, here's the oocyte, here are sperm cells arriving on the outside. You can barely make them out. They're wispy tails, right? So once fertilization happens, then the additional polar bodies will be made and meiosis II will be completed. But you can see already, this is the presence of a polar body over here. Okay. All right. So hopefully that really impresses upon you the two reasons why meiosis is so important, right? The fact that we sexually reproduce means we have to divide our diploid genome in half in order to only contribute one copy of the genome to our offspring, and the other copy comes from our mate, right? And two, just as important, if not even more, is that meiosis provides the opportunity for this mixing of genes, right, to form a unique new version of the genome. That is why your kid doesn't look, exactly like you or your partner, and likewise, that's why you don't look exactly like your mom or your dad, right? All right, let's go ahead and stop there.